Welcome everybody to Chem 313 Lecture 10, and we are going to be continuing today with our talk about biological membranes. And specifically what we're going to talk about today is going to be membrane proteins and also membrane transport, which involves an important class of proteins known as channels and transporters. So we are going to have to think about protein structure again for the purposes of today's lecture. So last time, to review, we said that membranes are bilayers of lipid, and specifically phospholipids and sphingolipids, usually mixed together. And we said that the reason that these lipids form bilayers is because of the shape of the molecules. The molecules have a polar head. That's the phosphate plus the head group if you have a phospholipid. And we have nonpolar tails, which are the fatty acids. So we have an amphipathic molecule, is another way of putting that. And if you were to draw a box around them, you would see that they have a rectangular, approximately, cross-section. So those are the properties that you need in order to form a lipid bilayer. So what happens if you mix these with water is that our lipid molecules will spontaneously self-organize into a structure that looks like this. With water all around. But by forming a lipid bilayer, the fatty acids can avoid contact with water, which is just the hydrophobic effect. All right, so what I'm showing here is the structure of cholesterol. And at the end of the last presentation, we said that cholesterol is important in, I'll say, modulating the fluidity of a lipid bilayer. Now you'll recall that fluidity means the liquid-like character of a bilayer. And we said that usually at a higher temperature, a bilayer is going to be more fluid, all else being equal, which stands to reason. So at the end of the last lecture, we drew this graph. We said that you can actually measure fluidity of a bilayer as a function of temperature, and what you would see is a graph that looks something like this. So that the fluidity increases with temperature always, but there's this very well-defined characteristic sharp transition that we call CHEM, or the melting temperature. And that corresponds to the temperature where the lipid bilayer essentially melts. You go from a solid-like gel phase, which is what dominates at low temperatures, to a more liquid-like liquid crystal phase, which is what's common at high temperatures. And the melting temperature is simply the transition between those. Now we said that this graph is for a bilayer composed of just pure phospholipid. So you have a phospholipid dispersed in water. no cholesterol or no other lipids at all. So we left with this question. We said that if you were to mix in a small amount of cholesterol into your bilayer, and remember that cholesterol will not form bilayers by itself, we get a different shape of our fluidity graph. We said that it starts off higher and the transition is kind of much more gradual, something that looks sort of like that. So what do we notice here? We notice that the transition is less well defined. It's more gradual with temperature. And the other thing that we note is that the fluidity in the gel state, which is the one that's essentially solid, increases, but the fluidity in the liquid state decreases. Right? We notice that the purple curve is shifted sort of down compared to the non-cholesterol bilayer. And the question I left you guys with last time was why? Why do you suppose this happens? It clearly has something to do with the way that cholesterol and the phospholipid molecules interact. So I'm going to explain this to you right now. There's actually two questions on homework 4 that are related to this, and so by walking you through this exercise, I think I'm kind of giving you guys a freebie. So, you know, I would attempt homework 4 before you watch any further in this video. But let's take a look at what happens. Let's take a look at a solid-like or a gel phase lipid bilayer. So we said last time that in the gel-like state of a lipid bilayer, and I had a diagram showing this, you'll recall that our fatty acid chains are mostly going to be extended, and they're going to be very closely packed together. So our bilayer is going to look like this. All right, so that's what a solid-like lipid bilayer would look like if you could look at the individual molecules. Now, what would it look like if you were to add a little bit of cholesterol? So you guys remember something that I said in the last lecture when we were talking about cholesterol. We said that if you were to line up a molecule of cholesterol next to a fatty acid, we said that the cholesterol is going to be shorter than the fatty acid molecule. It's shorter and it's also wide. Now, because of this OH group that's on the cholesterol, this is going to want to interact with water, of course. The rest of the molecule is nonpolar and is going to be trying to hide away from water. So if you were to add cholesterol to your lipid bilayer, what's going to happen? You're going to see a lipid bilayer that looks something like this. Bear with me because it's going to take a minute to draw this. What I'm doing is I'm drawing the cholesterol in red. And I'm drawing the cholesterol as a short sort of flat molecule with the OH group sticking out into water. So what did we just observe? Well, because the cholesterol is shorter than the fatty acid, but the OH group wants to be sticking up into water, when you insert cholesterol into a gel phase bilayer like that, you end up introducing these gaps between the fatty acid chains. So what's the big deal about that? Why are the gaps important? Well, if the gaps are introduced between the fatty acid chains, what does that mean? That means that the fatty acids are not in direct contact at that location. That means you have more free space between your fatty acid chains, which means you have more mobility of those chains, right? Because there's nothing in the way of a fatty acid molecule from diffusing or rotating to fill the space. And if you have more mobility, that means you have higher fluidity. That's in the solid-like phase or the gel phase. Now let's take a look at the liquid-like phase. Called liquid crystal or liquid ordered, or depends on which source you consult. So we said last time that in those lipid bilayers, our fatty acid chains are more spread out. They're not rigidly packed together because the chains are sampling different rotamers. So our chains are in less contact. We'll say they're disordered. So what would happen if you were to add cholesterol to this lipid bilayer? What would happen is you would see something that looks like this. Again, bear with me, I know it takes a long time to draw. So what do we observe as different in this case? Well, we said last time that what happens when you have very rigidly packed fatty acid chains like so is that introducing cholesterol, because it's shorter than the fatty acids, is going to introduce some gaps. What ends up happening here is because we have our fatty acid chains disordered and the whole membrane is kind of squished a little bit flatter, we start out with gaps between our phospholipid or sphingolipid molecules, and introducing the cholesterol fills the gaps. And this is exactly the opposite of the previous case here. So by filling in the gaps, the fatty acids are going to become more restricted, and therefore your fluidity goes down. So as a result, cholesterol, we say it modulates the fluidity of the bilayer. What we mean by that is it increases the fluidity of rigid membrane, and it decreases the fluidity of disordered membranes. And this is actually pretty important in living cells, because in living cells, remember, a lot of organisms are going to be subject to big changes in temperature. And if your lipid bilayers don't have any cholesterol in them, you can have a big change in fluidity with a fairly small change in temperature. So what's nice about cholesterol is it kind of helps to insulate membranes from changes in fluidity. That's important for the function of membrane proteins, which is what we're going to talk about next. So I hope this slide gives you guys some hints on how to do those couple of problems on homework 4. All right, so it's tempting to think of lipid bilayers and membranes as just these two-dimensional liquids of phospholipids and sphingolipids, which they sort of are. But a better way to think of lipid bilayers is a solvent for proteins. This is a more accurate picture of what a lipid bilayer actually looks like in a cell. It is completely packed with proteins. In some cells and in some organelles, there are membranes that are actually more protein than they are lipid. There's just enough lipid in there to sort of glue everything together. So this is kind of a busy diagram here. I think I want to introduce to you guys the concept that there's two different broad classes of membrane proteins. There's integral and there's peripheral membrane proteins. So we're going to talk about each one of these separately. Different textbooks differ on how they define or draw the boundary between an integral or a peripheral membrane protein. Here's how I'm going to have you guys learn the difference. An integral membrane protein can't be removed from the bilayer without somehow disrupting the bilayer using, like, detergent or some sort of organic solvent to dissolve away all the lipids. That's the definition of an integral membrane protein. And there's two different classes of that. There's transmembrane proteins, which is the most common one, and then there's a second type called lipid anchored, which we'll talk about both of these in a second. And then the other type of membrane protein is a peripheral membrane protein. And a peripheral membrane protein is just bound to the bilayer by weak intermolecular forces and can be easily removed just with mild treatments like changing pH or something like that. We're going to talk about peripheral membrane proteins first because, they're sort of the less important of the two. Not that they're not important, they're all important, but there's less to know about them. So peripheral membrane proteins, we said, are simply bound to the lipid bilayer by your regular non-covalent intermolecular forces. So that could be ion interactions or hydrogen bonds, usually with the head groups of the phospholipids. So our head groups are all polar. They're mostly hydrogen bond donors and acceptors. So side chains that are polar can form hydrogen bond interactions with these lipids. We can also have what's called an amphipathic alpha helix. I showed you guys an example of this in the protein structure section. If you were to look down the length of an alpha helix where you've got your R's pointing out along the sideway, you can end up in a situation where the R groups on one side of the helix are nonpolar and the ones on the other side are polar or charged. And in those cases, this side of the helix can just sort of lie down in the plane of the bilayer, sort of like a hose lying in grass, so that the nonpolar side chains on that side of the helix are sticking down into the fatty acid part of the bilayer. Some other proteins have what's called a hydrophobic loop, which is where you just have a short stretch of nonpolar amino acids that kind of dip down into the bilayer. And then the last category I don't think really counts, but this figure includes it, so I'll include it. And that's when it's bound to an integral membrane protein. So in other words, it forms some sort of quaternary structure with an integral membrane protein, which is one that goes all the way across the bilayer. So to give you guys more realistic pictures of what this would look like, NADH oxidase. This is an enzyme that is an amphipathic alpha helical peripheral membrane protein. Phospholipase C, a different enzyme. This one has a hydrophobic loop. It's actually got two of them right there and there. All right, integral membrane proteins. The most important class of integral membrane proteins are the transmembrane proteins. Transmembrane proteins completely cross the lipid bilayer. They poke through one side and stick out the other side. Now, this may seem kind of difficult. How could an integral membrane protein do this? Remember that the backbone of any protein is going to have lots and lots of peptide bonds in it. And these peptide bonds, of course, must form hydrogen bonds. But if your peptide backbone is buried in the lipid bilayer interior, there's not going to be any water around for them to hydrogen bond with. And they definitely cannot hydrogen bond with fatty acid chains because those are just hydrocarbons. They can't form hydrogen bonds. So the way that most integral membrane proteins solve this problem is they form a very long nonpolar alpha helix. So remember, in an alpha helix, the backbone of your protein coils up like this. The peptide bonds form hydrogen bonds that run down the length of the helix. So that satisfies the need for the peptide hydrogen bonds. So we have hydrogen bonding between our peptide bonds. You get that in all alpha helices, not just transmembrane alpha helices. But then we have our side chains sticking out all around the perimeter of our helix. And as long as these side chains are nonpolar, they can easily interact with the fatty acid chains. And there's no loss of energy because they don't have to hydrogen bond with anything. They're just comfortable being away from water. So in order to form a transmembrane alpha helix, we'll say here, transmembrane alpha helix, we need a long stretch of nonpolar amino acids. Typically at least 20 amino acids in a row that are nonpolar to give you a transmembrane alpha helix. This diagram is showing us what these transmembrane alpha helical proteins look like. Over here is a protein whose sequence is color-coded, where the orange amino acids are the nonpolar amino acids. And what do we notice? We notice that the amino acids that are sitting in the lipid bilayer are mostly nonpolar, with a couple of exceptions. So we see that there's threonines right at the edge of the nonpolar part, and that's because threonine's side chain, you'll recall, looks like this. So threonine you'll sometimes see right on the edge of the lipid bilayer because this part can stick up into water, whereas the methyl can point down into where the fatty acid chains are. So those are our two polar amino acids that we see in the transmembrane part. The other three are glycine. So glycine, you'll recall, the side chain is just hydrogen. So that's kind of a neutral amino acid. It's like not terribly polar, but it's also not terribly nonpolar. So this figure just counts that as a semipolar amino acid. But the important point here is that essentially all of the chains that are inside the lipid bilayer are going to be nonpolar. So you need to have a long stretch of nonpolar amino acids to cross a lipid bilayer. That last slide showed you what's called a single-pass transmembrane protein. This slide is showing you what are called multi-pass integral membrane proteins, which are proteins that have several transmembrane alpha helices. So, for example, this protein here, which is called rhodopsin, it has 7 transmembrane alpha helices. In fact, almost the whole protein is buried inside the lipid bilayer. This protein down here is a glucose transport channel. It has also several transmembrane alpha helices. But in this case, they surround an internal water-filled channel, which is very important for the function of certain proteins, and we're going to talk about that momentarily. So this is all I'm going to say about integral membrane proteins for now. A question I have for you guys is, do you think there are any transmembrane beta sheet proteins? Do you think that's something that's possible? The answer is yes, they do exist. They're not very common, but there are such a thing as transmembrane beta sheet proteins. See if you can figure out how that's possible. All right, I'm only going to give you guys one slide of our next type of integral membrane protein, which is lipid-anchored proteins. Lipid-anchored proteins are covalently attached to a lipid in the bilayer. So I'm just giving you guys a single example on this slide. What we have here is a case of 2 examples of a protein linked to a fatty acid. So there's 2 commonly found fatty acids attached to proteins. There can be a myristic acid, which would be 14,0, and that's typically attached to the N-terminus of a protein. Another one is palmitic acid, which is 16,0, and that one, when you see it attached to a protein, it's usually connected to a cysteine on a side chain. You don't need to know that. So the lipids that are commonly attached to proteins are either a plain old fatty acid. That's the most common case. So the fatty acid chain sticks down into the bilayer like here. The second thing is something called a prenyl group. I'm not showing you guys any example of that. That's essentially an isoprene chain. Remember what isoprene is. We talked about that in lecture 9. And that also sticks into the lipid bilayer just like the fatty acids do. The third class of lipids that can be anchored to proteins is something called a GPI anchor. And that's an entire specialized phospholipid. So the protein has its own dedicated phospholipid molecule that attaches to its C-terminus. I'm not going to draw a diagram of a GPI anchored protein because they're big and complicated, but the book talks about them if you care. I don't care that you know too much depth about lipid anchored proteins, just know what they are. Okay. Now that we've introduced the concept of membrane proteins, we have to talk about a very, very important concept for essentially all of biology, and that is transport across membranes. So if you have a cell, in order to survive, your cell has to be able to take in solutes from outside and expel other solutes that it doesn't need anymore. So for example, it may need to take in food sources like glucose, and to excrete waste products like, say, CO2. And so that means that these solutes, whatever they are going to have to cross a lipid bilayer. So do you think it's possible for these molecules to simply pass right through a membrane? That's the question I have for you guys. So obviously they get across the lipid bilayer in some manner, but as it turns out, some things can just pass right through in a certain direction. Others cannot. So to answer the question, can a small molecule pass through a membrane? Here's the short answer. Small nonpolar molecules like gases and to a lesser extent water, yes, they can cross through a lipid bilayer. So these small nonpolar molecules can pass right through, with the caveat that they must go along a concentration gradient. More on that in a second. Anything that is larger or polar, or especially anything that has an electrical charge, no, these cannot pass right through. So in these cases, transport across a bilayer requires, I put this in quotes, catalysis. And by catalysis, that's provided by proteins such as channels or transporters. So why is it that small and nonpolar things can pass through a bilayer, but larger and polar ones cannot? Well, if you look at a bilayer, what is a bilayer? We've got our fatty acid chains on the inside and our polar head groups of our phospholipids or sphingolipids on the outside. So if something is going to simply pass right through this bilayer, what it has to do is, through diffusion, it must encounter the lipid bilayer and then just under thermal motion work its way through the bilayer and then across on the other side. We'll say that this is side A and this is side B. So if you were to draw a free energy diagram for this process, what would it look like? Let's say we are going to draw the free energy diagram as a function of our reaction coordinate, and our reaction coordinate in this case is our solute on side A and our solute on side B. So let's say that transport in the direction of side A to side B is energetically favorable, so that side B is lower in free energy. What happens in between? What happens in between is we have to pass through this transition state. And what's this transition state look like? That's where the solute is completely buried in the bilayer interior. So why is the barrier so high? Well, if you have some sort of a polar substance, when it's over here on side A and when it's over here on side B, it can hydrogen bond with water, which lowers its free energy. But when it's inside the lipid bilayer, it's not hydrogen bonding to anything, and that raises the free energy, right? You also have to consider the fact that when it's in the bilayer, not only is there no hydrogen bonding, but you're in a sterically crowded environment. You've got to push your way through all those fatty acid chains. So that's going to be high in free energy. So really the only molecules that can cross a lipid bilayer like this are small molecules which have minimal steric clash, And they need to be nonpolar, which means that they don't mind the lack of hydrogen bonding. So for a polar solute like, say, glucose, this is what the free energy diagram would look like. If you had a nonpolar solute, I'll use a different ink color for a nonpolar solute. So if you had a nonpolar solute, for example, oxygen, its free energy diagram would look more like this. It's definitely going to be higher in free energy because of the steric crowding, but it's going to be much lower. So for that reason, only the nonpolar stuff can pass right through the lipid bilayer. All right, we already said that nonpolar and small solutes can pass through a bilayer directly. This is what's called passive diffusion or passive transport. Anything that is larger or polar or charged cannot. Okay, but regardless of which type of solute you have, whether it can pass straight through the bilayer or not, it will only flow across the bilayer in the direction that is spontaneous. So what do we mean by that? They can only flow in a certain direction, and that is in the direction with the delta G of crossing from side A to side B less than 0. So on the previous slide, we assumed that the delta G for crossing from side A to side B is going to be favorable, right? So the delta G of crossing from side A to side B less than 0 implies favorable. Favorable is another way of saying spontaneous. To cross from side A to side B. That does not necessarily mean it's fast. If you have a polar or a charged compound, it's still going to need a channel to cross because it's not going to be able to pass straight through the lipid bilayer. Now, if you had transit where the delta G of moving from A to B was positive, that becomes unfavorable, which means that if you wanted to go from A to B, you're going to have to do some sort of work. So what we have here is a case of spontaneous versus non-spontaneous transit. If you're trying to move a solute from one side to another of a bilayer and it is unfavorable, you have to do work to get it to happen. That's what's called active transport. Work must be done somehow. Oftentimes that's by using ATP, but not always. Now, if you're moving with the direction that the transit is favorable, if it's favorable or spontaneous to move from side A to side B, we have two other names for that type of transit. We can call that passive transit, which is the case in which the molecule just passes right through. Or we can have what's called facilitated transport. In which it is catalyzed. Now notice that for active transport, there is no possible situation where your solute would simply diffuse across the lipid bilayer in the direction of of unfavorable transit. That simply doesn't happen. And the reason that doesn't happen is because active transport basically requires the participation of membrane proteins. So we're going to talk about this in some depth. So what causes the free energy of moving from side A to side B to be favorable or unfavorable? By favorable, I mean less than 0, unfavorable, greater than 0. Well, if X is our solute, and we have a higher concentration of X on one side of the bilayer than the other, X is going to tend to flow from the side where the concentration is high to the side where the concentration is low. So it would be non-spontaneous, therefore, for X to go the opposite direction. So the most important factor in deciding which direction a solute will flow spontaneously is concentration. So generally speaking, the free energy of moving from side A to side B is favorable if the concentration of X on side A is greater than the concentration of X, on side B, right? So in other words, the free energy of moving X from side A to side B is going to be favorable if we move along a gradient, as we say. Okay, from high concentration to low concentration. And that's fairly intuitive for most people. However, I have to add an important note here. This is going to be true only for non-charged compounds. For ions, this does not necessarily hold true. So in this case, we can write an expression, an equation for the free energy, the delta J of moving from A to B is going to be equal to what? It's going to be equal to RT, natural log, concentration of X on side B over concentration of X on side A. So notice here, if the concentration of X on side B is less than the concentration of X on side A, then that means that the natural log term is going to be negative, and therefore the delta G of A to B is negative. So this is an equation that is essentially just the free energy of a chemical reaction. If you consider the chemical reaction X on side A becoming X on side B. Your equilibrium constant would be the concentration of X on side B divided by the concentration of X on side A, and then the free energy is just RT, natural log of the equilibrium constant. All right, so that works great if you have uncharged neutral compounds, but let's get into a sticky situation here. Let's say that you've got some sodium ions. We'll say that this is the outside, well, let's keep the same nomenclature. Let's say that this is side A and this is side B. And you got sodium ions on side B as well. So we see here that the sodium ion concentration on side A is greater than the sodium ion concentration on side B. So you would think that the delta G of A to B for sodium would be negative. But that's actually not necessarily true, because sodium is an ion. Why does that matter? I'll show you why that matters. Let's assume that in addition to all the sodium, we've also got some potassium ions floating around. And let's say that on side B, even though we've got very little sodium, we've got huge amounts of potassium. So do you think now it becomes spontaneous for sodium to move from side A to side B? Well, the concentration on side A is definitely greater than the concentration on side B. So that would tend to make transport in that direction favorable, okay? But the total electrical charge on side B is more positive than on side A, right? So it may look at, if you just look at concentrations, it might seem that sodium would want to go across the lipid bilayer in that direction, but it's going to be repelled by the excess positive charge. So what that means is that for an ion, you can't just look at concentration. You have to look at other factors as well. So that means that for anything with an electrical charge, our expression for the delta G becomes more complicated. This is the expression. Delta G of moving from side A to side B is equal to RT, natural log, concentration of X on side B over concentration of X on side A. It's always where you're going divided by where you're coming from, plus a second term, ZF, and then in parentheses, psi A, excuse me, that should be psi B, minus psi A. This is what's called the Nernst equation. And this term is oftentimes written as delta psi, but I prefer to write it as psi B minus psi A so that you guys can keep track of which direction you're supposed to be going. So what is all this stuff? Well, the first term is the same as we saw before. That's just accounting for the concentration of your ion. What's all this stuff over here on the second term? Z is the charge of the ion, including the sign. So for sodium, this Z would be +1. F is a constant that's called the Faraday constant. And it's equal to 96,500 joules per volt per mole, which is an awkward unit, I know. Psi B and psi A, these are the membrane potential, which is a fancy way of saying voltage, on sides B or A. So the psi is just a way of accounting for essentially the concentration of electrical charge on that side of the membrane. That's not actually what voltage is. Voltage is energy per unit charge, but you can think of it as the concentration of electrical charge on side B or side A. So if your psi B, let's say, is negative, that means that you've got excess negative charge on side B, for example. So the potential difference, the voltages on side A or side B, that's usually measured in volts. And so if you plug in volts for your delta psi, joules per volt per mole for Faraday's constant, the charge on your ion has no sign, you end up with an answer that's in joules per mole, which is standard units. So you do not need to memorize the Nernst equation. This will be provided. And typically the membrane potentials on either side of the lipid bilayer, those are going to be given to you. There's ways that it can be measured, but it's very complicated and it's really outside the scope of this class. So for any problem I give you, you'll just be provided with the delta psi. So you will have to do problems like this. Notice that if the charge on your ion is 0, so you in other words do not have an ion because there's no electrical charge, the whole second term goes away and you're left with the same equation that we saw earlier for the free energy of transit of an uncharged molecule from side A to side B. All right, so to summarize, passive transport. Passive transport is when the delta G of moving from A to B is negative and you have a small nonpolar solute. And that means it just passes right through the bilayer. Facilitated transport is the situation when the delta G from side A to side B is negative, favorable, but you have catalysis to speed up the transport. That means you've got a channel protein. We'll talk about that momentarily. And then lastly, we've got active transport. That's transport in the direction of delta G from A to B being unfavorable, and that requires not only catalysis, but work. Okay, so where does this work come from? So the work for active transport comes from either ATP or from what's called coupled transport. All right. So ATP we're going to talk about later in the course, but ATP is the molecule in which hydrolysis of ATP in the presence of water, giving you ADP plus a phosphate ion, releases energy. And this energy can make an otherwise unfavorable reaction favorable. What's coupled transport? Coupled transport is when you carry two or more solutes at the same time. Solute A has its delta G for transit being negative, and solute B has its delta G for transport being positive. So in essence, transit of solute B is sort of paid for by transit of solute A. So you move both solutes at the same time. One of them is favorable and the other one, the transit is unfavorable. And as long as the favorable one is more favorable, you end up with a net delta G that is negative. Now, what about the catalysis? For facilitated or active transport. That's done by this class of proteins called channels. Channels, for the most part, are transmembrane proteins, if this is our lipid bilayer. Where they provide a pathway through the bilayer so that the solute doesn't have to simply pass straight through. And that's important for polar molecules or for ions especially, because an ion or a polar compound, when it's in the interior of the bilayer, is not going to be able to form hydrogen bonds. And so the channels sort of act as an alternate solute, an alternate solvent for the solute while it's passing through the lipid bilayer. So let's take a look at channels and their closely related cousins, transporters. All right, channels and transporters. These are transmembrane proteins arranged around a central pore that let molecules or ions pass through. Most transporters and channels are specific, which means they only let one solute or a couple of closely related solutes pass through. So, for example, there's sodium channels which will not allow potassium to pass through, or other ions. So this diagram gives an example of a glucose channel. And in this case, we have our interior pore right here. And for this particular channel, there's actually a conformational change that occurs when the glucose binds to pass through. The whole thing just sort of inverts in and out so that our glucose can bind on one side and then pass out on the other side. So some channels, not all, can open or close. So for example, this voltage-gated potassium channel, this channel undergoes an allosteric transition from an open structure, which you can think of as being like the R structure, to a closed structure, which is something like the T structure. And you can see by looking at this channel, so this is supposed to be a top view of the channel, the difference between the open and closed conformations isn't that much. We just have all of these transmembrane alpha helices, and when you get the conformational change, they sort of slide together sort of like a camera shutter, so that in the closed state, this pore through the center is just small enough that the potassium can't pass through. In the open state, the potassium can pass through, but it's a very, very tight fit. So channels are not like an open hole in your membrane where you can just drop the solute right through. It's always a very, very tight fit to fit through, and that's important for reasons we're going to talk about in a second. So what's the difference between a channel and a transporter? So this is a kind of shifting sand situation. The definitions seem to be changing on this one. And unfortunately, like most things, they're getting more complicated. So the current definition of a channel is that they are for facilitated transport, and they may or may not be gated, which is a fancy way of saying open under some conditions and closed under other conditions. The key definition, or the key feature of channels that makes them channels, is that they're relatively simple. They bind to the solute, and the solute simply works its way through under thermal motion. That's what makes a channel a channel. A transporter, these are mostly for active transport. Transporters are complex. They involve large motions or conformational changes. And usually, not always, but usually involves some sort of a chemical reaction to take place. So for example, ATP hydrolysis. So an example of a transporter would be, for example, our sodium-potassium ATPase, which you may have heard about it in other classes. Now, when I learned about channels and transporters, the rules were a lot simpler. They told us channels are for facilitated transport, and that's usually the case. These are usually for facilitated transport. Transport being spontaneous. Transporters are usually for active transport, which is why they require things like ATP hydrolysis. So for example, the sodium-potassium ATPase, if this is the inside of the cell and this is the outside of the cell, what the sodium-potassium ATPase does is it takes 3 sodium ions from the inside of the cell and shoves them out, and 2 potassiums from the outside of the cell and brings them in. Now, the delta G of transport is positive for both of these. So how does the sodium-potassium ATPase do this? How does it move 3 sodiums out and 2 potassiums in when both of them are moving against their gradient? Well, you need ATP. So you have this big complicated molecule that undergoes these crazy conformational changes. I'm going to post a link to a YouTube animation that shows you how this thing works. Channels are much more simple. I'll also post a link to some YouTube simulations of channel proteins, and you can see the difference between these two types of membrane transporter. All right, so channels especially, obviously transporters, but channels even by themselves, they function very similarly to enzymes, and that's what I wanted to conclude this lecture with is the idea that a channel plus some sort of a solute, S, so if S is our channel, and S is a solute, and we'll say that we're going to move from side A to side B, these proteins behave very similarly to enzymes, right? So in order for the channel to catalyze transit of a solute across the lipid bilayer, the channel must bind the solute. So what happens is the solute and the channel bind to give us a CS complex. And the CS complex can then break down to give us the channel plus the solute. We're going to say that this is on side A and this is on side B. So what we have here is a situation that is very much like the Michaelis-Menten model for enzymes. And in fact, you can even use the Michaelis-Menten equation that you would for enzymes. So if this is side A and this is side B, and we've got a channel protein stuck in this lipid bilayer, our solute is going to pass through the channel from side A and come out on side B. So that means that the rate of our transport, or V, is going to be equal to what? Well, it's going to be equal to the change in the substrate concentration or solute concentration on side B with respect to T. And as it turns out, this V can be expressed by a Michaelis-Menten equation. Right? So we have the concentration of S on side A because that's where the solute must first bind to the transporter, is on side A. The Vmax is the maximum possible rate, which is what you see when all of the channels are saturated. And if you were to make a graph of the rate of transit as a function of solute concentration on side A, and we would have to make the assumption that the solute concentration on side B is very low, or that the whole thing is irreversible. But if you make those assumptions, you would find that a graph of the rate of transit would look very much like a Michaelis-Menten graph. And so the key take-home from this is that channels can be saturated. There is an upper limit to how fast a solute can pass through a channel. So again, channels are not just like a hole in a bilayer that you can take a solute and drop it right through. They're much more like enzymes. The solute has to bind, and then it has to work its way across, and there's only so many binding sites per channel, and so there's only a maximum rate at which solute transit can occur. So I'm going to say see the video links if you guys want to see what this looks like in reality. And in the meantime, take a look at homeworks 4 and 5. I will see you guys later.